Today we're going to be talking about Punnett squares. You might be asking yourself, what is a Punnett square? Well, this figure right here is a Punnett square. But what is this box for and why do we use it? Well, in science, we use Punnett squares to determine the possible genetic outcomes for the offspring. These outcomes are a result from the combination of the parent's genes. But how do we use this Punnett square? What does that really mean? Well, we're going to do an example together using guinea pigs to figure it out. In this example, we're going to have a dad with black fur shown up here up top and a mom with white fur. It doesn't matter what order you write these in. We just picked this one for convenience. Black is going to be our dominant gene in this example, so we write it with a capital letter. Now, if you ever see one with a capital letter, that means that it's going to be dominant, which means that it is expressed. So, that means that the animal is going to look or express whatever the dominant gene is. If we have a recessive trait, though, that means we have lowercase letters. For a recessive trait to be expressed, we need two of the recessive traits or genes. To complete the Punnett square, we combine the genes from the mom and dad by pulling the letter from above down and the letter from across over. So now we could fill in the whole Punnett chart doing this system. Now, we've completed the chart, and if we fill in the genes, we can see that if an individual has two different genes, we can say it's heterozygous. So, in this example, what percentage of the guinea pigs are going to be heterozygous? Well, like we just said, it has two different genes. Each of these guinea pigs over here look like they have two different genes because they have a capital and a lowercase. That means that 100% of them are going to be heterozygous, and they're going to be heterozygous black because it has a capital letter, which means that it is the expressed dominant trait. Now we're going to take a girl and a boy from this generation that we've just created in generation 1, and we're going to cross them for a second generation. So we set up our Punnett square. Remember, each of the individuals in the last generation were each heterozygous. So over here, we have a capital and a lowercase. So we took a male and a female. So we set up our Punnett squares. What different combinations could we do? We do the same technique that we used to write the top letter and bring the other ones across. And this is what we get. These are all the different combinations we could have. So when we find all of the gene combinations an individual has, we call it a genotype. And this is what we just found. So what colors could these guinea pigs be? What different colors would we see in this generation? Remember, in the last generation, we only saw black guinea pigs. Well, in this generation, we're going to see that 75% of them are going to be black and 25% are going to be white. And these are our phenotypes, which is what we see as a result from the individual's genes. Now remember, we know what color they're going to be based on if we have these capital letters or lowercase letters. If there's a capital letter, it's always going to be dominant, the dominant trait, which in this case is black. But if we have two of the lowercase letters, then it's going to be the recessive trait, which in this case is the color white. So now we say an individual is homozygous if it has two of the same genes. In this example, do we have any homozygous guinea pigs? We have two. We have this individual, which is homozygous black, and this individual, which is homozygous white. So 50% of these guinea pigs will be homozygous, but only 25 will be homozygous white and 25 will be homozygous black. Do we have any heterozygous individuals? Remember, that means they have two different genes. Yes, we have two heterozygous black guinea pigs. Congratulations, now you've learned all about how to use Punnett squares, and if you have any questions, feel free to go back, rewind, and rewatch this video again.